Inquiry is something you are going to experience hopefully a lot this year in this chemistry course. If you look up inquiry and try and find its definition, you may see things like this. Inquiry means a seeking for information by asking questions. Or it may be defined as interrogation, or a question or questioning. It would involve a search for truth, for information or knowledge, examination into facts or principles, and research and investigation. We engage in inquiry in chemistry every time we seek an answer to a question. The scientific method is the process that we follow when engaged in inquiry. Observation involves the use of your senses to obtain information. Many times observations lead to a question being asked. We can then use any available information to create a hypothesis to be tested. Sometimes we are presented with a question and we have to make observations before developing a hypothesis. Once formulated, a hypothesis is used to make a prediction of what will happen under certain conditions if the hypothesis is correct. Now we come to the experimentation part. Scientific experimentation is the act of testing hypotheses. To create a valid test of a hypothesis, in other words, to create a valid experiment, all factors that can change, these are called variables, so all factors that can change during the experiment must be identified ahead of time. The experiment must be set up so that only one variable is purposely changed. Everything else that can be is held constant. The one that is purposely changed is also said to be manipulated. The manipulated variable is also known as the independent variable and is purposely changed to see how other factors change in response. A factor that changes in response during the experiment is called the responding variable and is also known as the dependent variable. What would be the independent and the dependent variables in the experiment described in this picture? As we look at the picture, we see that it says how light affects plant growth. So it looks like we're going to be checking the growth of the plant as we change the amount of light. So we are purposely manipulating the amount of light that the plant gets. So that's the manipulated or independent variable. And we are going to check to see, maybe by measuring plant height, how much the plant grows. So that would be the responding variable or the dependent variable. We would have to hold other things constant. Things like the type of soil things are planted in, the type of seeds that we're planting, the amount of water that we give them, etc. We'd want to keep all of those things identical between all of our potted plants as we checked how light affects each one. When performing an experiment, detailed notes on the setup and control of the experiment, in other words, the procedures followed, must be recorded. The results of an experiment are only valid if they can be obtained consistently from repeated performances of the experiment. So people need to know what you did to get your results so they can see if they can follow those same procedures and get the same results as you did. The results of an experiment may be quantitative or qualitative. Now let's look at these two terms. Quantitative changes. These are things like counts, how many of something there is, or measures. These would be recorded in data tables. From this data, charts or graphs might even be generated. Qualitative changes might be recorded in a data table. Things like color changes, whether a reaction occurs or does not occur. Or these changes might be written down as just short notes, a summary of your observations. Now this slide on the left shows a couple of different tables presenting quantitative information collected during an experiment. You can see that they all involve numbers because they're numerical quantities, counts, or measurements. On the right-hand side, we see a qualitative table. The information is presented in a table like the other was. However, this information does not involve numbers. It involves descriptions, either verbal or pictorial. So these are qualitative observations. They don't involve numbers. This is a picture of a graph that might be generated from your quantitative data. It gives you a picture, a way to visually represent your data and look for trends, like this data appears to fall nearly in a line. So it would be approximated by a line. And so you might want to generate something like this as you go to interpret your data. It makes the interpretation process go more smoothly. After an experiment is performed and all data is gathered and organized, the gathered information must be analyzed. What is data analysis? This is where you interpret the information and compare it to the prediction you made based on your hypothesis. So does it support, meaning go along with your prediction, or not? Then you draw a conclusion, but what's a conclusion? An experiment should always end with a summary of what was done and an explanation of how the results do or do not support the original hypothesis. What do you think? Do experiments fail? If the results don't support the hypothesis, does that mean the experiment's a failure? No. It may mean that you need to revise your hypothesis or write a completely new one. But you now have more information upon which to base your new hypothesis, so you have more information than you first began with. So it is not a failure. Sometimes you may need to look at your experimental setup and see if it was faulty. Allowing more than one variable to change at a time can cause results to look like they do not support a hypothesis that's really true. So you do want to look at that type of thing. But an experiment is never a failure. Even if you have to go back and change the way you had it set up, you've learned something in the process. Now, if there are doubts about the methods you used, it is important to identify sources of error or things that could or should have been done differently. These should be reported at the end of your experiment as well, so anyone wanting to perform further experiments can improve upon your experimental design. After a lab report is written, is that all that there is? It's usually shared with others in the scientific community. In other words, communication is important. Traditionally, experimental results were shared through letters among colleagues in a society or through publications of results in scientific journals. Today, those things still happen, but results may also be shared through e-journals, email, teleconferencing, and maybe even be presented at international conferences. The most reliable information still comes from published works. Why? Why does it matter whether it's been published or if it's just something sent out by a scientist in an email or on an e-journal? There is a reason. Published works must first undergo a review process called peer review, where other experts in the field look for errors in their experimental design or maybe challenge the conclusions that have been drawn from a well-designed experiment. Take a moment to look over the peer review process that is depicted in this slide. The peer review of journal articles can be painful for a scientist, but is good for science as a whole because work that is not well founded is usually not published. Some journals publish preliminary results on the internet because the review process can take from 6 months to a year or even longer. Preliminary results should be treated with caution because such results are subject to change. Remember that no one knows everything. So collaboration is something that's very important in any science. Collaboration increases knowledge, skills, and resources available for problem solving. Each person's mind may approach a problem from a different point of view, especially when collaboration includes multiple disciplines. It is not without its problems. Great minds do not always think alike, nor do all scientists play well together. But its benefits can greatly outweigh its drawbacks.